This week we're going to talk about the concept of reproducibility and how it applies to reliable knowledge. Reproducibility is sometimes also referred to as replication, and it's the ability of an entire experiment or study to be duplicated and to obtain the same results. I like to stick with the term reproducibility because replication is a term that we use when describing having multiple experimental units in a treatment. And so it could get confusing, but you will see it referred to as replication in the literature, even some of the readings I've given you. And, if you think about going back to middle school, you probably all were taught that reproducibility is one of the foundations of the scientific method. In other words, if someone does an experiment in their lab and comes up with a certain result, the idea is that if another lab repeats those same methods, that they also will come up with the same result. So it really is a foundational principle, and it addresses the reliability of science because we all want to find relationships or hypotheses that are repeatable in all different circumstances. Otherwise, our knowledge is not particularly helpful. Recently, many scientists have suggested that we have a reproducibility crisis. And this is a results of a survey that was done and published in the journal Nature. And they got over 1,500 researchers were surveyed, and over half of them suggested that there was a significant reproducibility crisis. In other words, that there was many studies that could not be reproduced. This idea of there being a crisis started back in 2015 when some funding was available for psychological researchers to reproduce studies that had been published in some of their top journals. And when they did that, only 36% of those publications' results were found to be similar in the study when it was reproduced. Another study, which I haven't given you the readings for, but you could take a look on your own, in cancer biology, found that only 10% of the studies could be reproduced. And that's cancer biology. That's really kind of scary. You are assigned this paper for reading this week, but just to summarize what Nosek et al. Found is, like I said, they were given some funding to repeat 100 published studies that had been published in some of the top psychological journals. 97% of those studies had significant effects. They funded different laboratories to repeat those studies following the same protocol, and the results were really surprising. Only 36 of the repeated studies found significant results. The mean effect size of the repeated studies was half of the original studies. And they did something really interesting. They looked at what factors might predict whether the repeated study was similar to the original study. And it was best predicted by the effect size. In other words, how large a difference there was between the treatment and control groups. It was not predicted by the characteristics of the research teams. And these are characteristics of, number of publications that the various scientists on the publication had, or their citation frequency in the literature. So basically, even if you look at papers published by the quote bigwigs in psychology, but I'm sure this would be true in other sciences too, that's no guarantee that those results are reliable. And here's a summary of the outcome of all those reproduced studies. And you see the solid line here is the expected line if the effect size in the replication is the same as the effect size in the original study. And here they use replication instead of reproduction. And you can see that down here where there's small original effect sizes, the effect size in the reproduced study also tend to be small, and a large proportion of those are not significant. As you move to the right, such that the original effect size becomes larger, there still are some non-significant effects when you reproduce it, but a much higher proportion of those are now significant and similar to what the original effect size had been. And you can see another thing here with these probability plots along the top and the right side of this figure that just about all of those original studies, as I mentioned, had significant p-values. That's indicated by the blue. There's a little bit of gray here indicating non-significant. And when you look at the reproduced studies, you have a high proportion of those that were non-significant. Of course, their effect size is clustering around 0, and a small proportion that were significant and their effect size is larger. So this brings up the question of why is there such a big difference between the original and the reproduced study? Well, one major effect might be publication bias. Scientists only submit papers when results are significant, even if they are just barely significant. That seems to be the currency of whether or not many scientists decide to submit a paper. And that can lead to a bias in published literature. And we know from talking about type 1 and type 2 errors, there are a certain proportion of situations where you will find a significant effect, a type 1 error, when in fact the null hypothesis is not significant. But still, you would only expect that to happen, maybe 5% of the time or so if we set our alpha at 0.05. So what else is going on here? Well, oftentimes you can take data that maybe don't show much of a difference and tweak them a little bit to get a significant result. One approach is to remove outliers, and this is something that statisticians commonly encourage scientists to do, but it can be taken too far, especially if those outliers might then make what was a non-significant result now a significant result. A lot of times scientists will transform their data. If you have data that are severely non-normal, you can do log and other kinds of transformations to try to make them more normally distributed to fit the assumptions of some of the statistical tests. But that can also then perhaps have some causing bias towards more significant results. Also, including or excluding covariates in the analysis may make the variable you're interested in, or the dependent variable you're interested in, become significant or not, and that's not something that's easy to discern from reading the methods. There could be just a tendency to suppress the non-significant results, maybe not talk about those in your paper as much and focus on those few significant effects that come through. And again, perhaps biasing towards situations where overall there's really not much going on, but you can focus on one or two aspects of it and say, well, this is interesting. There's another approach that's sometimes used called optimal stopping strategy, where, maybe to save money, you um, start a study and you continue it until the results are significant. So you just keep adding more data, collecting more information. If you're working with people, as many of these psychology studies are, you just keep adding more individuals to your study until you get the significant result, and then you stop. Well, that's going to bias you towards getting significant results, even when those may not be. And finally, there are many situations where somebody will say, well, the p-value is 0.056. It's, just a little bit bigger than 0.05. And so we still think that's interesting and that it should be considered. Well, the more you do that, the more you're going to kind of creep into this area of tending to publish results that really are not statistically significant. Lindsay, 2015, came up with what he referred to as the troubling trio. So things that should make you skeptical of the results in a particular scientific paper. The first was low statistical power. That can come from either low sample size or a great deal of variation. We've already talked about factors that influence statistical power, so I won't go into that. Secondly is what he referred to as a surprising result. So something that wouldn't be expected. And in that case, maybe you would want to have, a really, really robust data set to make sure that the results that you're seeing is something that's real. Couple examples I can think of in this situation in wildlife biology. One is the result that was found both in Yellowstone National Park and now has recently been found in Nevada as well. Situation where in Yellowstone National Park, grizzly bears were coming into the dumps. This was back in early days, up until the early 70s. The National Park Service had big landfills within the park where they dump all the food waste and so on from the hotels and restaurants. And grizzly bears found out very quickly that this is a great big food resource, and they'd come in, and in fact they even had bleachers set up around some of these dumps so that people could come and sit there and watch the bears come and eat and take pictures and stuff. And surprisingly, when they removed the this food source, this very abundant food source, initially there was a decline in bear numbers, but over time the number of bears actually increased in the population until now it's higher than it was before the food was being supplied. And you think about that, that's surprising. Take away food and population increases. But there was a long, very long monitoring of this population. The approaches were very robust, and I think everyone agrees now that this has occurred, and now we've seen it also happen with black bears in Nevada, probably because these food sources attracted bears but also resulted in very high mortality, because once bears become accustomed to human food sources, they often start causing trouble and getting into homes and cars, and then they have to be euthanized. Long description there. But the third is p-value close to 0.05, and obviously for the reasons we've talked about, that could easily get you into trouble. If you look at the published literature, another thing you find is that most studies are not repeated. In other words, it's very difficult to find a publication that basically just repeated exactly what somebody else did, with the idea of showing that it's reproducible, even though, as we talked about, that's really one of the foundations of science. There's a number of reasons for this. One is just lack of resources. Secondly, there really are very few incentives to publish positive replications. In other words, just again, as I said, publishing something that somebody else already found and saying, hey, I found it too. A lot of journals, they have very limited journal space, and for them, that's just not a high priority. On the other hand, journals are also reluctant to publish negative findings. So, if you come up with a cool idea and you go out there and test it and it's not significant, to be true to your science, you really should put that out there and say, hey, thought that this hypothesis might explain, what's going on here. And well, we tried testing it and it was non-significant. Journals just don't, again, tend to publish that kind of thing. And the last one I think is a biggie, and that is that there's a premium placed on new and innovative discoveries. So the Discovery Channel is called the Discovery Channel, not the let's reproduce somebody else's work channel. There's a premium put on finding new, cool, interesting stuff. And scientists are, attracted to that. And so they tend to not want to just go back and repeat something that's already been done. So given that there is this problem of reproducibility, what do we do about it? Well, this was posed to these same scientists in the Nature study that I cited earlier. And there was three things that really came up consistently. Over 90% of the respondents saying these would be important things to do. One was to give scientists a better understanding of statistics. So really addressing some of those troubling trio issues. Secondly, better mentoring and supervision of graduate students that are becoming scientists, to teach them how to do good science and really publish those studies that are showing strong effects, and maybe not focus so much on those that aren't. And the third, which actually combined has the highest proportion that got that were indicated as important by these scientists, was more robust study design. So that's why you all are taking this class, knowing what constitutes a good study: replication, randomization, controls, treatments, and so on. A lot of scientists really don't get that fundamental understanding of how to put together a good study. And so that's why I think courses like this are so important. And I'll let you take a look at those other ideas that are listed there. There has not been a similar study done as has been done in psychology and cell biology, where somebody has come up with a big pile of money and said, hey, go replicate all of the studies that have been done in the Journal of Wildlife Management or the Journal of American Fisheries Society or so on. And so we really don't know, how big of a problem this is in our field, but I would actually argue that it's more likely that this is a problem in ecology, natural resource management than in, say, psychology. And that's because we have some special challenges, right? We know that there's high natural variation out there, which um, tends to mask effects and make it more difficult to find those important effects. We know that, even though you may find an effect of a certain plant species on deer um, health and reproduction in one area, that may not apply in another area. There may be a whole suite of different plant species that they can feed on. So that could affect whether or not that difference is found. Same thing, obviously, in aquatic environments. Temporal differences, so droughts, wet periods, and so on. An effect that's found in one situation may not be found in other situations. Doesn't mean that it wasn't real when they measured it in one time period, but changes in resources may make that more or less important in another situation. We know that there are many, many factors that influence response variables, and it's very difficult to control all of those factors, making, again, reproducibility more difficult. And finally, funding is tight. So again, this idea of, why would you go and just repeat an experiment or a study that somebody else has done when funding is limited? Generally, your agencies are going to say, well, I want you to go tell us something new and different. We think we've already figured that one out, even though maybe you haven't. So what's the bottom line from all of this? Well, basically, if you look at the two studies that have been done recently, the majority of the published studies out there cannot be reproduced. I'm not saying that that means that they're absolutely wrong. Could be, as we talked about, temporal spatial differences that might mean it was perfectly correct that they found an effect when they looked at it one place, but just isn't important in another place. But that definitely makes you concerned about the reliability of that particular result. As we talked about, be especially skeptical when there's low sample size, surprising result, p-value close to 0.05. Effect size is the best measure of reliability. So even if there's big names on the publication, that doesn't mean that that's more likely to be true than the publication of some first year grad student who just happened to find some really big important effect that nobody else had seen. And bottom line is, don't believe everything you read in the published literature. I think there's a tendency for people to think, gosh, it was published in this journal, Nature, Science, JWM, whatever it might be. And, these people are really smart and they do good work, and so it must be right. Well, not necessarily. Listen to Pauline here and question things. Use what you're learning in this course to really question the literature and come to your own conclusion as to whether you will believe or not believe the results of a particular study.